Hello, everyone. Biological membranes are basically the way of maintaining a cell's architecture and physiology. And in this session, we are going to focus on one of the key components of the membrane, and that is the membrane lipids. And in this session, even more so, we are going to focus on that membrane lipid which is abundantly present in the bilipid layer, and that is the phospholipids. So let us look at the learning outcomes of this session. The lipid bilayer that is dynamic, it is ever changing, constantly changing. And the bilayer confers exceptional physical properties to the cell. So one of the membrane components that contributes to both of these, that is the dynamism of the bilipid layer and to the physical properties, are the phospholipids. So phospholipids are amphipathic. That means that they have a end that can be hydrophilic and one other end that can be hydrophobic. So they are able to interact with non-polar components, and they're also able to interact with polar components. And it is this amphipathic nature that is present in the phospholipids that enables them to form spontaneously a bilipid layer. Moreover, they constitute, as mentioned earlier, the bulk of the lipids in the membrane. They are either glycerophospholipids or they are sphingophospholipids, with fatty acids and phosphoric acid being the major constituents. Apart from having a very structural effect, phospholipids contribute to membrane permeability, signaling, membrane transport, etc. Now, let us focus on understanding how phospholipids' basic structure makes it an amphipathic molecule. So, let us first look at glycerophospholipids and then we will look at the sphingophospholipids at a later stage. So the glycerophospholipids contain three or comprise three components. One is the glycerol moiety, and this glycerol moiety forms actually the backbone of the phospholipid. So you can see how carbon 1, 2 and 3 are associated all with OH groups. One has, other than the glycerol, the phosphoric acid moiety, and the third component is the fatty acid itself. As you can look at the fatty acid itself is amphipathic, with one end, that of the carboxylic end, being polar, while the other end, which ends with a methyl group, is non-polar. So this long chain fatty acid is having its own amphipathic nature. Now, let us consider the glycerol moiety in such a way that you have the two OH in these two carbons positioned over here and this OH positioned over here. Now, the fatty acid would try to interact with this OH in an ester bond. Why? Because this is a alcohol group while this is the acid group. And we know that if you have a carboxyl end and a enol group, then together they can form a covalent linkage which is called as the ester linkage. So at this point, a removal of H2O will lead to formation of a ester bond. So you have therefore a C double bond O formed between the first carbon of the glycerol with the fatty acid. The same will happen with the second carbon OH. You would have an esterification between the fatty acid and the second carbon of the glycerol moiety. Now, when you have just one fatty acid attached to the glycerol moiety, then this is what is called as the monoacyl glycerol. When you have the second fatty acid also attached to the second carbon of the glycerol, then this forms what is called as the diacyl glycerol. When you have on the third carbon the OH, H being substituted by a phosphoric acid, then now what you have is a phospholipid. So very clear indications of the fact that when you have the polar group or the polar end of the fatty acid in an interaction with the polar end of the glycerol, this entire portion is now not available to interact with any polar constituent. What is available is the CH3 end at this point. What is available over here is the CH2, and both of these ends are going to be non-polar, so they cannot interact with water, so they are hydrophobic. Okay, same goes with this. Because of the esterification, the polar end of the fatty acid is no longer available to interact with hydrophilic components, but it has only the hydrophobic end which can interact with hydrophobic components. Unlike these two, in the third carbon, on the third carbon where you have the phosphate moiety, we know that P is attached to oxygen. Okay, you have the double bond O, you have O minus, you have O minus, and so this becomes a highly polar group and hydrophilic at the same time. So if you were to draw the structure in this manner, you would have two hydrophobic tails, okay, and you will have one hydrophilic head. So this is the hydrophilic end, this is the hydrophobic fatty acid chain, and this becomes the hydrophobic end. So these are the two hydrophobic tails, and this is the hydrophilic head. That is how a phospholipid is always represented in books, where this is the hydrophilic polar head, while these are the two hydrophobic tails, and which are nothing but the fatty acids that are bound to the carbon number 1 and carbon 2 of the glycerol moiety. So that is very clearly indicative of why the phospholipid is an amphipathic molecule. This component, the head component, makes it hydrophilic, while the tail component makes it hydrophobic. So within one structure, you have a hydrophilic moiety and you also have a hydrophobic moiety, and therefore together it becomes an amphipathic molecule. Now let us consider that you have a vessel in which there is water, and into this water you are adding different phospholipids. Okay, and you mix these very well and then let it be. And after you let it be, what is observed is that all the phospholipids align themselves such that they form a monolayer. Okay, monolayer of what? Monolayer of phospholipids. So interesting therefore, and very expected is the fact that all the hydrophilic heads are going to align in such a way that they are interacting with the water because they like water, while all the hydrophobic tails which do not like water, they are hydrophobic, so they are directed away from the water. And that is how this composition or this structure is stable, because you would find that the hydrophobic tails would never want to interact with water, while the hydrophilic tails would like to interact with water. So spontaneously they will be aligned in such a way that they attain a kind of stability. Now suppose you were to push this entire monolayer down into the water phase, then what is immediately going to happen is, because the hydrophobic tails do not interact or cannot interact with the water, again the entire monolayer is going to get dispersed into a non-monolayer. So you will have all the phospholipids again in solution all mixed. Okay, and what will again happen is that this will come back to this monolayer formation. So this is something that is happening spontaneously. Let's again consider a pool of water, or basically an aqueous environment, and let us put the monolayer in this. Like mentioned, this monolayer is going to be unstable. Why? Because this part of the monolayer is definitely not liking to interact with the water around. So ideally what is going to happen is, if there are more phospholipids present, you would have another monolayer being formed, and this monolayer will have its hydrophobic tails interacting with the hydrophobic tails of this monolayer. So now you have immediately the formation of what is called as a bilipid layer, and this bilipid layer is going to be definitely more stable than a monolipid layer within the water environment. So as such, therefore, what you have very clearly observed is the formation of a bilipid layer by the phospholipids with with complete understanding that the hydrophilic ends will be interacting with the water phase, while the hydrophobic tails will be present interacting with each other to get into a stable conformation. So this is definitely thermodynamically favorable. Now let us ask the question as such. When you look at this, do you think this is completely stable? So the answer to this is it is not completely stable because these portions of the bilipid layer are still exposed to water on these sides, and that water is going to definitely affect the hydrophobic groups that are present over here and present over here. So you will find that there is a lot of instability at the ends of this bilipid layer, and hence it is not completely stable. Therefore, if these ends of the bilipid layer interact with each other, like you see over here, there are no free ends. So none of the hydrophobic groups are actually interacting with the water. It is only the hydrophilic ends over here and the hydrophilic ends over here that are interacting with the water. So very clearly what you observe is compartmentalization, and the compartmentalization has actually separated the inner water layer from the outer water body. So you effectively have this portion now that you can consider as intracellular. So you must understand that this is equivalent to being a liposome, okay? Or you can consider it basically as a membrane, okay? So this becomes intracellular, this becomes extracellular. And this becomes the protective barrier, which is the bilipid layer formed just by the phospholipids. And because phospholipids have the or they are amphipathic, they are semi-permeable. So you must understand that water can still go through the bilipid layer, but the rate of the diffusion of the water across can be decided by the composition of the bilipid layer per se. Moreover, small molecules like carbon dioxide, oxygen, they can also traverse this bilipid layer, and hence this is a semi-permeable membrane. Now let us go to looking at the fact that there are several glycerophospholipids. Okay, so we did understand that you have a glycerol backbone of which the first carbon and the second carbon are associated with the fatty acids through an ester linkage, and the third carbon is attached to a phosphorus moiety, but the phosphorus itself can attach to an X group. Now if the X group is a serine, then this is what is phosphatidylserine. If the X group is choline, then you have the phosphatidylcholine. If this is ethanolamine, this is forming what is called as phosphatidylethanolamine. If this is inositol, it forms what is called as phosphatidylinositol. Plus, if you have two phospholipids linked to each other through an X group, then that is what is called as diphosphatidylglycerol. And this is effectively cardiolipin, which is found in a lot of bacterial membranes as well as in the inner membrane of the mitochondria. So you must understand that all these different types of glycerophospholipids can be present in a bilipid layer. It is not that a bilipid layer is made up of only one phospholipid. It can be made up of all these different types of glycerophospholipids. So let's say consider this is phosphatidylcholine, this is phosphatidylserine, this is phosphatidylethanolamine, and this is phosphatidyl inositol. Okay, you can consider any of them to be anything. My point is that each of them is a different glycerophospholipid. Now, say for example, you have a bilipid layer that is made up of only one glycerophospholipid, then what is very distinctly observed is that if you have a line of symmetry here or a line of symmetry here, you will find that this bilipid layer has a certain amount of symmetry if you consider only the head groups, correct? We still do not know what is the composition of the fatty acids over here. We only know that the head groups are same, and with respect to the head groups, there is a kind of symmetry that is there, or an axis of symmetry is possible. Now let us again consider, suppose you have these four glycerophospholipids and you have the same set of glycerophospholipids arranged such that a particular phospholipid is facing the same phospholipid again on the other leaflet. If you consider this to be a bilipid layer, then what you would definitely observe is that if you have an axis of symmetry here, you will not find that there is a symmetry across this line. But if you have if you have a line drawn over here, you will find that there is a symmetry across this. So in such a case, with respect to the heads of the glycerophospholipids, one line shows asymmetry while the other line shows symmetry. The question is the bilipid layer arranged in such a manner? So what is observed that there is no rule that it has to be existing in this manner. So if you have the phospholipids present like this on the other leaflet, it is absolutely not necessary that a phosphatidyl inositol should only have a phosphatidyl inositol in this leaflet, exactly opposite to it. So you will never really find that the composition of glycerophospholipids in the outer leaflet is same as the composition or the distribution of the phospholipids in the inner leaflet. And therefore, if you draw a line of symmetry this way, there is going to be no symmetry, so there is asymmetry. And if you also draw a line of symmetry in this manner, again you will find that there is no symmetry. So effectively what we are trying to understand is this, that there are different types of glycerophospholipids contributes to the membrane being asymmetric, because the distribution of the phospholipids is already making the outer leaflet very different from the inner leaflet, and so there is no symmetry between the two, and so effectively the entire bilipid layer is asymmetric. Now, therefore, we have to say that distribution of the different phospholipids in the bilipid layer is asymmetric. Now, say for example, we so far had looked at the head group. Now, let us say for example, consider the fatty acid chains that are present in the phospholipid. Now, say for example, one of the fatty acid chains is longer than the other fatty acid. Okay, you can already see a certain level of asymmetry over here. Suppose this is facing the same phospholipid with the head group being the same. But then look at the difference in the lengths of the fatty acids that are present in this phospholipid and the length of the fatty acid that is present in this phospholipid. So you can already see that between this and this, there is a asymmetry clearly present. If you add another phospholipid, well, this may have symmetry, but you still have overall asymmetry because of the length of the fatty acid. So what we are therefore trying to understand is that asymmetry in the bilipid layer can also be because of the different lengths of the two fatty acids chains in a glycerophospholipid. So you may have a C16 fatty acid, or you may have a C18, or you may have a C20. So you can have different lengths of fatty acids attached to the glycerol of the phospholipids present, and that itself is also going to contribute to the asymmetry of the bilipid layer. Why are we focusing so much on the asymmetry of the bilipid layer? Because the Singer-Nicholson's fluid mosaic model had one of these proposed characteristic, and that is saying that the bilipid layer is asymmetric, and that it is asymmetric contributes to its diversity, contributes to its functional diversity and structural diversity as well. Now we look at one another aspect of the fatty acid chains in the phospholipid per se. So what has been observed is that most phospholipids that are part of the membranes have one fatty acid bound to the carbon number 1 of the glycerol moiety, and the other fatty acid bound to the carbon number 2 of the glycerol moiety, with of course a third bound to the phosphoric acid, and the phosphoric acid bound to a functional group which could be choline, which could be ethanolamine, serine, anything. Now, it has been observed that generally in phospholipids that are present in the bilipid layer, you have one fatty acid saturated and the other fatty acid is bent. Now, the reason that it is bent, so you have a kink, you can very distinctly see there is a kink. Now, the reason for this kink is because this fatty acid is unsaturated. So, in a phospholipid, one fatty acid is generally saturated fatty acid, and the other fatty acid is generally an unsaturated fatty acid. So, because it is unsaturated fatty acid and the double bond that is present is a cis double bond, you have an effective bend formed over here. So, what happens because of the formation of this bend in the fatty acid? So let us say, for example, consider a bilipid layer that is comprising phospholipids that have fatty acid chains which do not have any kinks. That is, they do not have any unsaturated fatty acids. If that is the case, then what is observed is that the membrane is going to be highly compact because they are all aligned in such a way that there is a kind of compactness that is present. Now let us look at the fact that one of the phospholipids in one of its chain has a kink. Okay, so you can immediately see that there is now a space between these two phospholipids thanks to this kink. So what is very apparent is therefore that with the presence of unsaturated fatty acid, now you can imagine this one has the unsaturated fatty acid. What if this one has? What if this one has? So between every phospholipid, the space is going to increase. And that is contributing to the membrane becoming less compact or it becoming more fluidic because the phospholipids have space for themselves to be present within the bilipid layer. So you must definitely therefore understand that the presence of a cis double bond in a fatty acid is contributing to the rigidity and the fluidity of the membrane. And not just that, it is also contributing to asymmetry. So you can see while you have this over here, here you do not have it. So again asymmetric. Here you have it, here you don't have it. So again asymmetric. So the asymmetry is also because of the kink or because of the presence of an unsaturated fatty acid in the phospholipid. So by far, therefore, what we have looked at is that the head region of the phospholipid definitely contributes to asymmetry. The length of the fatty acid also contributes to the asymmetry, but the presence of saturated fatty acid or unsaturated fatty acid also contributes to asymmetry, and that in fact also contributes to whether the membrane is compact or whether the membrane is less compact. So it is definitely affecting the fluidity of the membrane. So phospholipids contribute to the fluidity of the membrane because of such characteristics present within its structure. Now let us look at another thing, and that is that at different temperatures, okay, depending on the composition of the bilipid layer, you may have the membrane existing in different phases. Say, for example, you have an optimal temperature, which means a cell that is residing in a optimal temperature. So then the cell will have a bilipid layer that is a kind of liquid crystalline phase. So it is not completely solid, nor is it completely liquid. It is kind of semi-liquid or semi-solid. Now, suppose you increase the temperature, then what is going to happen is that due to an increased temperature, the membrane will become more fluid. Okay, that is something that will easily be observed and that leads to hypoosmolarity. While if you uh, if you subject this cell which is in optimal temperature to lower temperatures, then what is going to happen is that the membrane will become more rigid, they will become more compact, the movement or the fluidity of the membrane is going to decrease, and so you will have what is called as hyperosmolarity. So you can see how the membrane lipids are contributing to the osmolarity of the cell. So another functional aspect that is very, very important to be considered with respect to the biological membranes. But one statement that is very important over here is this, that membranes can transition from one phase to another when and if required. So to understand this, therefore, one question that comes up is, so membranes are two-dimensional fluids? Is it or is it not? Now, why is this transition from one phase to another phase important? Because depending on the residing temperature, the membrane composition will have to vary. So suppose you had in this condition less of unsaturated fatty acids, then it would become compact faster. The transition would be much faster. If you would have more of saturated fatty acids over here, then in this case the transition would not have been that faster. So you can see how the composition of the membrane is going to invariably affect the rate of transition from one phase to another phase. So, to understand this a little more, let us ask the question, will microbes residing in Antarctica, that is freezing conditions, have a different membrane composition than those residing in hot sulfur springs? The answer to that is yes. The composition of the membrane in Antarctica will be very different from the composition in hot sulfur springs. And where is the difference? The difference mainly is in the length of the fatty acids and also in the saturated and and the unsaturated fatty acids present. So, if you have unsaturatedness present in the membrane structure because of the presence of cis double bonds in the fatty acids, you will find that there is uh, more fluidity observed over here. And so, if you transfer this membrane to a lower temperature, it will take a longer time to freeze, or it will take a longer time to become rigid, or it will take a longer time to become solid. But if you had a membrane that contained only saturated fatty acid, you can already see that it is highly compact. So when you move to a lower temperature, you can immediately understand that this is going to very rapidly form a very compact structure, a rigid structure, a structure which has frozen. So, is this good for the microorganism that is growing in Antarctica? No, because if this is frozen, then how is it that the membrane interacts with the outside and the inside? How does it allow any kind of interaction between? And if that is the case, then the cell is going to die. So if in the Antarctic region you have membranes which are more like this, then there is a more there is effectively more interaction that the membrane can do with the environment outside and the environment inside. So you can see how the membrane composition is important in making the membrane fluid and rigid. And that is a contribution by the phospholipids. So, which is why it is very important why the phospholipids are an abundant lipid present in the bilipid layer. Now let us go to the second type of phospholipid that is present in the bilipid layer, and that is sphingophospholipids. So in the sphingophospholipids, you basically again have a structure that is going to give rise to a hydrophilic head and to a hydrophobic tail. Please do understand that you have another R group over here, which is a long chain hydrocarbon. So you would have another phospholipid, or sorry, you would have another hydrocarbon chain over here. So you'd have two tails and you'll have one head, which is very similar to the glycerophospholipids itself. But the composition is slightly different because you have the parent or the backbone here not being glycerol. The backbone over here is what is the sphingosine. And the sphingosine is associated with an inherent an inherent hydrocarbon chain, and the second fatty acid chain is associated with the NH2 group of the sphingosine, while the OH group of the sphingosine is interacting with the phosphoric acid, which in turn can interact with another X group. It could be ethanolamine, it could be choline, it could be anything. So therefore, you again can see very well that you have two hydrophobic tails associated with the backbone, and that associated with the phosphoric acid, and phosphoric acid associated with the functional group. So this becomes the hydrophilic head and this becomes the hydrophobic tails. This is what is sphingomyelin, and this is present in the mammalian cells. In a lot of cell types, sphingomyelin is definitely present. So you are witness to the fact that this too is an amphipathic molecule, and hence it is going to be present in a stable bilipid layer. Now let us look at one another aspect of phospholipids is that what is the distribution of the phospholipids? Is the distribution of the phospholipids going to have any functional effect, or is the phospholipids moving around in the bilipid layer? So we already know that Singer-Nicholson in their model proposed that the lipids and the proteins in the bilipid layer are in constant motion. Okay, so let us look at whether phospholipids do have some kind of motion associated with themselves. So very clearly what has been observed is that the phospholipids are associated with several different motions. One of the most common motion is what is called as the lateral diffusion. So you can have one phospholipid moving either this way or this way or this way or this way. So you could have this moving in all the four axes in a plane. Again, you must understand that if this phospholipid is moving in this direction, it is not necessary that this phospholipid has to move in this direction. This phospholipid can move in the other direction. Okay, but so therefore, what is understandable is that lateral diffusion is a motion that the phospholipids definitely are observed to have. The second observation is that in its place itself, the phospholipid can rotate. They can spin in that place itself. They can just keep rotating, 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 rotating. So that is a rotatory motion that is associated. Moreover, the tails of the phospholipids can also flex, and they can kind of move around. So all of these can kind of move around like this. So there is definitely a motion set within the bilipid layer thanks to the phospholipids. So therefore you can already see how there is dynamism present in the membrane, because these motions are constantly happening, and these constant motions are going to lead to the dynamism of the membrane. There is one other motion that is observed with the bilayer, and that is what is called as the flip-flop motion, but this is the rarest kind of motion that has been observed, and it is also cost effective. So when we it is also costly or expensive. What do we mean by that? It means that if you want a phospholipid to move from the inner leaflet to the outer leaflet, then it has to be driven. So there are several enzyme systems which are responsible for enabling the movement of phospholipids from one leaflet to the other leaflet. It goes the other way around also, from the outer leaflet to the inner leaflet as well. One of the examples of such an enzyme system is what is called as flipases. So the flipases enable the flip-flop movement, and you can have the composition of the outer leaflet with respect to the inner leaflet changing based on the flip-flop movement that can happen. So that is important. That is, composition of the inner and the outer leaflets vary with respect to what is the phospholipid that is present. What is the distribution of the phospholipids that is present in the two layers. Another interesting facet about the phospholipids is that they themselves have a very intrinsic structure or shape, and that depends on the type of head group that is present, and also the length of the acyl chain, that is the fatty acid chain, and whether it is saturated or unsaturated. So all these three components, or all these three properties of the phospholipids entails it to have certain intrinsic structures or shapes. Say for example, phosphatidylcholine and phosphatidylserine are cylindrical lipids. So whether you see the lipid here or you see the lipid here, they are one shape, okay? But you can have several other phospholipids that have a conical shape. So for example, phosphatidylethylamine, cardiolipins, phosphatidic acid, diacylglycerol, these all have conical shape. Okay, and the conical shape is because the head group is much smaller, okay, compared to what is present in PC and PS. So therefore you can see how they have different shapes based on what is the head group, what is the length of the fatty acid chain, and whether they are unsaturated or saturated. Interestingly, what has been found out is that in mammalian cells, uh, the phosphatidylcholine and sphingomyelin are concentrated more in the outer leaflet, while the inner leaflet has more of ethanolamine, and phosphatidylserine. It is only in cases where the cell is to undergo death that one has observed that the phosphatidylserine is flipped to the outer leaflet. So you can see how under abnormal conditions only phosphatidylserine has gone out. Otherwise it is present more in the inner leaflet. So again you can see that the distribution of the phospholipids is going to confer a different function to the outer leaflet and that to the inner leaflet, because the composition of the inner and the outer leaflet is very different from each other. Now, what is also interesting to note is that the presence of a certain type of phospholipid in a membrane bilipid is able to spontaneously lead to the membrane getting curved. Now, say for example, you have only cylindrical lipids, that is, you have only phosphatidylcholine and phosphatidylserine present, then you will find that the membrane is always going to be a flat membrane. Okay, but say for example, you have inside the inner leaflet having more of phosphatidylethanolamine, its head group is less. Okay, while the phosphatidylcholine that is present on the surface, its head group is large. So you can see how it has conical shape. So you can see that because the conical shape is such, you will have automatically the bilipid layer bulging to form a kind of curvature. Now, is this important? Yes, it is important because it can enable several functions like budding off of vesicles or budding in of vesicles. So, endocytosis or exocytosis. These phenomena are made possible because the membrane can get curved. Okay, it can also get curved depending on the surrounding that is present. Okay, so therefore you can see how PC being present on the outer leaflet and PE being present in the inner leaflet helps to curve the membrane. So this is something that is important. Suppose you had it the other way around, okay, where you have the conical face of the lipids present inside and that present outside and that present over here like this. Then in this case you would have an inverted curve, or you would have a reverse curve that is happening. So curvature of the membrane is also influenced by the distribution of the phospholipids in the bilipid layer. So let us look at what are the different points with which we can conclude this session. Both glycerophospholipids and sphingophospholipids are amphipathic, enabling them to be part of a stable bilipid layer. The head group, the fatty acid chain length, and the presence of the double bonds in the fatty acids contribute to the rigidity and fluidity of the membrane and as well as the asymmetry of the membrane. The phospholipids have shown to exhibit motions such as lateral diffusion, flexion, rotations, and flip-flop movement. It is interesting to note how the distribution of phospholipids leads to spontaneous membrane curvature, which in turn helps in several functionalities of the cell such as movement, endocytosis, exocytosis, etc. So phospholipids are not just the major structural lipids, but they are abundant and they are inherent structures that enable them to contribute to the charge on membranes, the fluidity, the shape, the asymmetry, and the movement within the membranes. Thank you.